Okay, so we're going to get started. It's a real pleasure to introduce Dieter Jaeger on his return to Ann Arbor. Dieter actually met Dieter not only about we started our careers in science not only about the same time, but also pretty much in the same place, which would have been in the old neuroscience building, which would have been in the basement of that building, which would have been about 4 stories below where we are right now. In those days, Dieter was a graduate student in Wayne Aldrich's lab, and I was a postdoc across the hall with Ann Young and Jack Penny. Dieter came to Ann Arbor from Tübingen. He's, as everyone here knows, I think Dieter's originally from near Frankfurt in the Rhineland. And then subsequent to completing his PhD with Wayne, he went to Caltech, where he was a postdoc with James Bauer and started doing computational work, and then went to Emory and is now the professor of biology at Emory. Dieter's also associated with the Udall Center at Emory, which has been a very successful research program on, and has worked extensively on the basal ganglia and its many connections, including work on cerebellar physiology, thalamic physiology, both at the cellular and more integrative levels. A really interesting combination of doing, very sophisticated electrophysiology and computational modeling. And he's going to talk to us today about some Parkinson's disease related research. So thanks for coming.
Well, thanks so much, Roger, for that great introduction. Yes, it's a great pleasure to be back at Giving Michigan always. I actually still donate every year to your graduate students.
Oh.
Hopefully it's going to be good news.
It's probably need more now than
I can see that. Fortunately, yes. So today I'll actually talk, it's very much about preliminary work in the spirit of our Udall Center Exchange. It's really ongoing work in our Udall project. It's not a finished story, but it's what we're currently doing and thinking about. And we adopted this new mouse model, which actually was not written up in our grant, but we learned about it just about when we had submitted that grant. And I thought it was actually a better model than what we had put in the grant, which was bilateral straddle injections, which we tried out a bit, but seemed to have very variable behavioral events. And this one looked more interesting. So we switched to this and took us a couple of years. And I'll pretty much talk about what we learned in those couple of years of implementing that mitopark mouse model. And I'm not here on a mission that this is a great model, but I think it's a very good model. I only talked to a couple of folks and said that maybe this is what you want to do. It does come with a fairly substantial footprint in terms of cost and just waiting for mice to be ready for work, but when they are really good. So let me introduce that mitopark mouse model first in a little bit more detail, because I'm sure not everyone knows exactly what these mitopark mice are and why we want to use them. So it is a genetic model of progressive dopamine depletion. And how do we get that? We get, and it was first actually implemented by a Swedish group, Ekstrand et al. From the Larsen group in 2007. We're using almost the same model, although not the one that is imported from his original strain. We are using it based on two substrains available from Jackson Labs. So you have to buy 2 strains of theirs and start breeding them in a combination, then you get the mitopark mice. But for like $500 a mouse, you can buy the 2 parent strains from Jack's, which is a lot easier than getting something from Sweden. It also turns out that there's a very small difference in the timeline compared to this strain. So people who work with the original strain may experience slightly different timelines than the ones that we see. So what is it? Well, you're crossing mice that have a Cree expression in all neurons that express dopamine transporters. That is, of course, the compact substantia nigra compacta in the VTA, but it's also some stray neurons in the retina, the olfactory bulb, and the gut. Interestingly, some of those are affected in Parkinson's disease as well, and we can get some of those non-motor Parkinson's symptoms because those neurons also will degenerate in these mice. So it's all dopamine neurons. And the other part of it is what is the Cree-dependent expression of a mutation is a mutation in the TFAM transcription factor, which is a mitochondrial transcription factor. And it's a knockout, so this is not dysfunctional then in those dopamine neurons selectively. And it leads to a slow deterioration of mitochondrial function, which then starts hitting the metabolism of these dopamine neurons, and they slowly deteriorate in kilo. And that works beautifully. According then with the depletion of dopamine neurons, and first the terminals go away, as we'll see, and then the neurons, we also get progressive then development of motor symptoms, but also some non-motor symptoms. Some of those may be a bit unique to mice, but some of those resemble non-motor symptoms of Parkinson's patients as well. It's been since 2007 on a slow but steady, and now I think rapidly increasing number of labs scheduled to use these mice. And there's now, the word mitoparkinson is used in, according to Udall Scholar, in 1,330 papers today. That's, I mean, there are tens of thousands who mention 6-hydroxydopamine. It's still the majority model, but it's fairly well on the rise. This is a review paper from 2021 by Beckstead et al. And it sort of summarizes what we know about the time course of these mice as of that time. And they are trying to see, so this is a Parkinson's patient in years, and then the mice is compressed. Instead of decades, we're talking months. But you get a similar progressive decline, and you actually still totally, even though the mutation is there from birth, you actually you don't induce this. It is there from birth, but the first symptoms really come maybe at age 8 or 10 weeks, and then more and more symptoms appear, as opposed to the toxin models, which sort of have a rapid onset, maybe then a little bit of recovery, and then they are sort of at steady state. Although personally, I would say the steady state is not actually happening after a few days, and that there's actually a lot of long-term changes, even in 6-hydroxydopamine lesion animals, that are compensatory. And for some of the complex physiological changes, I would certainly not look 7 days or 8 days after 6-hydroxydopamine lesion, but that's a side element. So here is the timeline of various dimensions of what happens in mitopark mice. One dimension, of course, is what happens to dopamine neurons and their physiology and their health. And one is a whole animal behavior, and then the anatomy of dopamine neurons. In fact, you only sort of TH staining is not a super sensitive method, and to get a significant decrease, it is only at about week 12. And this is also what we see, that we don't see any depletion before that. And then sometime later, the cell bodies will become scarcer in the compactor, although we never see them totally disappear, but we're only going to week 30 at most. And the behavior, interestingly, starts more with some cognitive deficits very early, actually. And I'm not quite, we just talked about that a little bit in my individual meetings, that some of that seems to be almost before you see significant TH depletion. And it's a little bit, in that sense, a mystery how that comes about. The motor symptoms track the TH depletion in the stratum more clearly. And you can see it in open field, and rotarot is sort of the typical thing that folks measure as a first approximation. So that's sort of the overview, and I don't what we do about the changed gene product and nuclear protein expression. This is more for folks who look at the molecular etiology of this. And honestly, I don't think it's a great model for studying molecular dysfunction in Parkinson's disease. So it's not there to look at cellular biology of Parkinson's disease. I think it's really a model for the physiologists to use. It's kind of the opposite of the alpha-synuclein overexpressing mice, which I think might be of interest to people who work on the molecular mechanisms of neurodegeneration, but certainly should not work on the neurophysiology side of things so much. All right.
Do they keep eating and drinking stuff?
Well, yes, but they don't live all that long. So they would die sort of at week 34, 35, and we have not tried to, at that moment, then to keep hydrating them. That might be partly because they don't eat enough anymore and they lose some weight. So they get to be pretty stationary at that time. But until week 30, they're perfectly fine with food intake and water intake. But after that, I wouldn't vouch for that. But I don't think I haven't seen a paper of someone trying to really push that envelope and keep them alive with visual food or intravenous nutrition or just sub-Q nutrition. And by the way, it's great to ask questions. I love interactive seminars. So please have any question. You might have to actually say them because I can only look at the slides, unfortunately. So here's our own TH staining results. So the 8-week is the mitopark. Here's a control. These are not taken from the same batch of staining. So it's TH is often best compared to control that is done in the same exact solution because it can show up darker or less dark in successive treatments. And this is what we actually do when we do 19-week and 24-week-olds. We do have, it's not shown here, but we do parallel staining of a litter cage mate that wasn't positive to have the exact comparison. That's how we then score statistically the TH depletion. But interestingly, it's also depletion starts more dorsally and around dorsomedial, dorsolateral. So it's not totally even, and it actually spares a little bit more of the ventral stratum initially, which is also similar to Parkinson's disease in many ways.
So is there any anterior to posterior gradient?
Good question. We have not looked at that carefully. If you think that would be interesting, we can certainly look.
Yeah, it might be pretty, early behavioral.
Okay.
Again, some of that are dorsolateral versus dorsolateral differences.
Okay, we can actually accumulate these over multiple mice, and it's an easy picking to look at more vectors and to quantify that. We have the slices. Actually, I should mention this right here is that this is the strength of the Udall Center, that we have a histology core run out of Adriana Galvan and Yolande Smith's labs and their histology center, which has done mouse anatomy and primate anatomy for decades. And they run for us all the TH staining and we get it back. So that's a real nice feature, and we don't have to train people in my lab anymore to do TH staining. Which, as a side effect, they don't know how to do it when they get my lab any longer. They used to. So I'm going to talk about some of these. These are all motor behaviors we do in the lab with these mitopark mice. Rotarod is just a standardized test. We don't do any, we're not planning to do any physiology with that. It's just a behavioral test. Open field, we do actually also use for some physiological assessments. They are twice weekly. They're just put in an open field arena for 15 minutes. We just do video of that to track their overall locomotor behavior and other behaviors. We do a trained water reaching task. That's sort of an interesting way to teach mice for forelimb reaching where they don't grab food pellets, which is a little harder to deliver food pellets one after the other, and then they may not eat 200 of them. So this water droplet reaching is a very stable reaching behavior, and they can, and we'll see a video, they can drink water out of their paw like it was a piece of food. And one can train them to reach it with their forelimbs. And we do some trained treadmill locomotion, but I won't be talking about that actually.
Peter, is the accumbens also lost in the VTA?
In the later stages, I think around week 30, yes. We have not followed that carefully either. There are among those 1,330 studies, I think you'll find one that would describe that. And we have to look for it. And it's also in that sense that the VTA is a little bit more resistant, even if it has the same cellular, and here we guarantee you it has the same cellular mechanisms at work, which we also believe, I think, in Parkinson's disease is the case, but it just sets it slower. And I think there are multiple different ideas. Some of them have to do with intracellular physiology and calcium current, but some of which has to do with the axonal complexity of these neurons, and the more axonal arbors they have, the easier it is for them to be metabolically challenged. I'm not sure anyone has proved one or the other idea as the ultimate one. That seems to be. These certainly have, the dorsolateral striatum has very extensive axon arbors. And it is the terminal fields that goes first. So it makes some sense that it's the metabolism of mitochondria and the terminals first that sort of makes it go bad. So here's a rotor rod performance. In our hands, they do actually more or less the same as controls until about week 16 on the rotor rod, and then we see a pretty steep and steady decline in the mitopark mice. And this is just time on the rotor rod. It's a classic rotor rod task. They just fall off after that time. Turns out that even if you do rotor rod for the first time, we find that it's not quite as easy as just popping them on there and see whether when they fall off. You have to look a bit carefully. Some mice develop strategies to cling onto the rod and swing around the rod with themselves, and you have to exclude those measurements. So one has to be a little bit aware of cleaning up these data from strange mouse behavior. So mice always find different ways to deal with certain situations, and you have to figure out what to do with those cases. These are the animal numbers on top. So just because we have the same cohort, but not necessarily tested every single half week, we put the numbers for each measurement point on top. This is the only time, sorry, I will mention gender differences, which is there aren't any that we could see in the motor behavior. And then we combine. We do both male and female mice, but we combine all the other data. And there was no difference here in the behavior on the rotor rod. So open field, we do a little bit more with open field. This is just a video session of their body position over 15 minutes of time as a function of age. And you see a fair amount of exploration and running around in the 10-week and 12-week olds. And then the mitopark mice sort of starting maybe week 14, 16, it becomes less and less trajectory, less and less forward locomotion over the time of 15 minutes until it becomes pretty scarce at week 24, 26. They move much, much less than they did. So we quantified this in some more detail. So first of all, the total distance traveled, you go from about 50 to about 18 or something like that. And these are 8 mitopark and 7 controls, and it's very consistent change. So motor, and every single mouse has that. So it's, in that sense, it's a good model. Interestingly, locomotion speed does decrease significantly, but not that much. How do we do speed? We're looking for periods or what we call a bout of locomotion, where they actually walk with the body speed being about a certain threshold, and then we score that as a bout. And while they're walking only a little bit slower, not significantly, but not a huge amount slower. The time they're in locomotion is much reduced, and then the last one is the number of periods where they are walking is maybe the way that really explains most of the immobility is just a lack of walking altogether. And they're also doing less rearing and other interesting behaviors. So they're much more likely to just sit there quietly, do nothing. And while this can be interpreted, I think, many different ways, one way that comes to mind is just a lack of initiation of movement in a purely Parkinson sphere of thinking. This is hard to disentangle from motivation, but it just is like a lack of initiation. And we'll see that in some of the other behaviors that I think they do have a problem with initiation. We do actually, so we wanted to know more about the locomotion and the step cycles. So we do a full reconstruction of their stepping. Our open field is a little bit unusual in the sense that it's a clear bottom open field, and we put a camera below. So we get a video, and we used to do 50 frames per second. We're just now going to 150 frames per second. But even with 50, you can get a pretty good idea of their individual paw placements. So the video can actually see each paw as it hits the plastic and when it lifts off. And you can use deep lab cut. I guess everyone is familiar with deep lab cut, I take it. This is really taking over the world by storm. this machine learning to track body parts with key positions, and you have, in our case, a very eager undergraduate students label something like 100 sample frames, and then the machine learning algorithm learns how to track every single frame, which I remember with Wayne, we developed fairly good GUIs to do it manually, but you still have to click on every frame. Now you only need a few sample frames, and then everything else goes automatic, which is kind of cool. But it does make mistakes, so you actually have to then correct all the outliers and the problems that it runs into. It doesn't work quite as flawless as it appears from the publications. And when you talk to people, it doesn't work flawless for anybody. You just don't see it in the publications quite that much. So we get the four-limb positions of all four paws over time. And we also track the center of the body, and then we get these kinds of plots of profiles of XY position that we can then triangulate and calculate the speed in centimeters per second for the body and then the individual paw. And so this is, in this case, it's just the hind paws, left hind paw and right hind paw. We see the alternating step pattern, and what we do for the locomotor bout, we give a threshold, which is this dashed line. When the whole body speed exceeds the threshold, then we score it as a locomotor bout, and then we can get the individual locomotor cycles, and we can, by crossing the threshold for the paw, this is when the paw starts moving. So that's what we call paw up. When it comes back down, it's paw down. And then you have the time in between, which is classically the swing time. And then in between that, you have the stance time. And that's how you can analyze locomotion. And you can now see what is going to go off in Parkinson in the mitopark mice with age in terms of the stepping cycle. So that's what we did. If you look at the stepping cycle, you have, here's the step interval. That's just the time between two successive steps. If you look at the mitopark mice in red, you see that over time they have a longer time between steps. So the stepping time becomes slower, which is also what we'd expect from a Parkinsonian type of condition. The swing proportion gets to be less. So what that means is that they spend more time in stance, pretty much. So less part of the step cycle is in swing phase. Interestingly, this is sort of just something we did and wasn't expected, but the stride length at the end of the day in centimeters doesn't really change. So each step still reaches the same forward amount of space, even though the swing proportion has changed. The swing itself is probably not that affected. It's just they stand a little longer, then they swing forward and get the same step size. One interesting aspect is a significant difference in the coefficient of variation or regularity of the stride length. And here the mitopark mice, which are in red, are showing actually a lower value for CV, which actually means that they are stepping in a slightly more regular way. And you can interpret that multiple ways. You could see it as a more stereotypic type of stepping pattern, and in a way maybe less flexible, less adaptive. Maybe they're a little less exploratory and looking left and right and doing individual steps. So it's a more stereotypic stepping pattern becomes a little bit more regular. That is somewhat a side interest of what's happening there. Now, one thing that we've started to do and actually pushing harder this summer and in this coming fall is electromyography, is EMG recordings from mitopark mouse model forelimbs. We are using a novel type of EMG electrode, which conveniently is being developed next door by the Sober Labs, Dr. Sam Sober, who is usually studying songbirds, but he was interested in getting EMG recordings from the vocal muscles of birds, and that didn't work with traditional EMG electrodes. So he teamed up with the faculty at Georgia Tech to make these silicon. This sort of rings true of Michigan, because when I was a grad student here, we actually were in the first phases of the neural network silicon probe development, and we almost got one to work, but never actually quite did. they couldn't solve the connector problem at the time. And it was very slow in coming. And now I see a similar kind of development with the biomatrix silicon probes that are there to implant in mice, but they actually also have human trials that I think are based in Canada, and they look at EEG recordings in humans, primates, and mice with slightly different probe designs. So we get to insert the probes into forelimb muscles. You thread all this all the way back to the skull, and then you put the omnetics connector, which is kind of big, onto the skull. And then you have a 32-channel EMG recording array. The hope is that this remains stable over the whole 10 weeks that we want to follow mitochondrial mouse. Unfortunately, so far, we're doing at best 2 weeks of stable recordings, and then one way or the other, one of the leads breaks or something goes wrong. So we're still trying to optimize the duration. And otherwise, we have to do many more mice and start the recordings at different stages of the disease. You can still do it, but it's more work. So we're trying to increase our duration. We can get very nice forelimb muscle EMG while they're doing open field, or for that matter, while they're doing the reaching behavior. And we can look at EMG activity. So that was Eli Lapesa in my lab did his honors thesis as an undergraduate in this. And now he's actually accepted to the Emory Georgia Tech Biomedical Engineering PhD program. So he's going to continue doing similar work going forward. He's interested in sort of prosthetic device development in the future. So that's cool, kind of. Then I don't have the slide, but we do the typical kind of bulk EMG. We rectify and smooth this, and then we have the smooth rectified EMG trace, which is what people generally do with EMGs. Then what we do is what we call a step-triggered average. So let's say we have a triceps recording. So this is the right triceps, and we average that to the event at 0, which is taking the right paw up. So this is the right triceps in relation to right paw up events. And maybe not too surprisingly, there's a burst of activity in the right triceps right before the paw up event. And we can quantify this burst by its timing and its amplitude. And this is then what Eli did. He, first of all, Z-transformed this. So this is our actual activity in arbitrary electrical units, or maybe microvolts. It doesn't really matter because EMG recordings are actually fairly variable in that. And we transform it to Z-scores. So that's standard deviations away from the baseline. And then he can make a confidence interval of where it leaves the baseline and score the onset of the EMG burst that is associated with each paw-up event, and the offset of the EMG burst, the peak time and the peak amplitude of that EMG burst. And we can see what happens with the triceps as mitochondria progresses, is the idea. This is actually this thing. This currently, it says 6 and 8, which is the number of recording sites from a single myomatrix implant in a single mouse. So it's one mouse, one control, and one mitochondrial mouse. So I wouldn't take there's statistically significant effects in this mouse, but you need, we are working on many more mice to make this more robust. So I would take all of these actual significances with a huge grain of salt. But we do see a lot of differences in the EMG between these two mice. So this is now the step cycle EMG burst duration. This is how long the burst is lasting. And if you look at the mitopark duration, it's firstly hugely more variable, and it's also shorter. And it's much more similar for multiple recording sites in the control mouse, and it has a longer duration. And if you look at the peak, we're looking at peak magnitude, that is the absolute peak size, and we are looking at the time of the peak. You see that the peak magnitude is down, and the time of peak is later. So that and that might explain a little bit the increased stance duration, because if you have this late triceps peak, there might be the way to pick up the paw this seems to be challenged. But again, a lot of more data are needed. Ultimately, because we're also trying to figure out a lot about the corticospinal pyramidal cells in motor cortex, and there is a fairly direct connection to motor units in the spinal cord, which this represents, we can hopefully get some correlations between cortical firing and this kind of change in the muscle properties for that particular population in Parkinson's. So the idea is to do it simultaneously with neurophysiology in the cortex. We're not there yet.
How much of a direct corticospinal tract to motor neurons is there?
Well,
because I think of that as being much more of a primate thing, having a strong innervation of motor neuron.
there is a good amount of interneuron interactions.
Right.
There's also some motor neuron, even in primates, it's primarily interneurons for that matter.
Right. I mean, it's a relative difference, but
Yes.
still, it is a significant difference.
There is not as much. It still is going to affect even the interneuron thing. Then will affect the timing of. Maybe not directly, but it will still impact the timing of motor neuron discharge in the spinal cord. So it's just with one extra interaction in between. All right, one mouse. All right, trained water reaching. So this is pretty much we have for the imaging task and also the coming neuropixels recordings, we have a head post, and the water reaching is done in a head fixed mice. We developed actually this very robust head fixation post that really optimizes stability of the tissues so that we don't get jitter of imaging data, or for that matter, instability of neuropixels recording. And the stability of this head post actually matters a lot for the quality of data you guys see. And it took us some iterations, and there's already 2 or 3 other labs that have started copying our posts, so they seem to be pretty good, comparatively speaking. This is just one picture of the mouse drinking a piece of water. There'll be some movies. I also wanted to point out, I really like this hardware. I fully recommend it. It's very inexpensive. It's called the BPOD system. It came out of the International Brain Collaboratory, which you might have seen papers from. And it is a microcomputer setup that is a state machine. It has a MATLAB API, so you only have to program your behavioral task instead of stimulus response. It's a real-time machine that will state by state advance the behavior through expected feedback from the animal, and then it advances to either failure or success states, or this cue or reward delivery. It operates. It has connections to a bunch of hardware like valves and aircrafts and LEDs and stuff like that on the input side. Sorry, that's the output side. It can get some digital and analog inputs, and it's only like it's increased from $700, I think, to $1,100 now, but it's still cheap compared to a lot of the other stuff. And it's very easily programmed with MATLAB, and it saves all the data that everything that has happened in terms of events on this machine gets automatically stored. No matter what you do with MATLAB, it will automatically store it as a HDF5 hierarchical file. So you can't lose any metadata of this machine, which is really nice. So fully recommended. The company that sells them is SandWorks. I also fully recommend it. Great customer support. It's totally open source. SandWorks has all the circuit diagrams, and then you look at all that and say, well, I'll rather pay the $1,100 than build this myself. Sorry, Wayne, because I learned how to build everything yourself at some point ago, but I've lost that trade in the meantime. So our behavior baseline, and they are not supposed to leave the touch bars. So these touch bars have a contact sensor. If they lose touch with the contact sensor during the baseline, they have to restart. That actually is one of the harder parts for a mouse. They do not like holding still, and they learn to fidget back and forth while not losing touch with the touch bar. But they're not happy holding still when they're anxious or nervous about an oncoming task. Then we have a sound or light cue, and depends on which mouse. We have used both. It's another side story how it took us some time to optimize what we do. So we started with the light cue so that we have multiple animals in neighboring boxes that can all be trained without hearing each other's sound cues. But now we also jumped on the train of measuring pupil diameter as a measure of arousal, which actually in our hands also works really well and is a good measure of arousal. And it can be done with deep lab cut. You can track pupil diameter and in a red light situation, so without really exposing them to daylight. But the light cue throws a monkey wrench in that because they will constrict the pupil when they have a light cue, and then that messes up the arousal measurements. So that's why we went from light to sound in our later cohorts. So now we can do the arousal undisturbed. Then they do the water reaching after they get the water. And then we have a consumption phase because it takes them some time to drink, as we'll see in a moment. And then is the intertrial interval where in the success trial, it's 2 seconds. If they don't move and don't drink, they get an extra period of waiting. So both if they fidget in the baseline or they don't drink, they get but as a form of punishment, they get a longer intertrial period. So to try and encourage them to perform with it. This is just an example. So here's the mouse. Here you see the liquid coming out actually, and then they grab the liquid. In this case, it takes them several times to get the whole drop. And drinking it, then wait. There's a drop. There comes the right hand. The left hand drinks the mouse.
So is there any paw preference?
I will show you some. Yes, there can be. It depends on the mouse. Some of them are mostly symmetric. There's almost a little bit of a preference for everybody. We initially tried to encourage them to use both by emphasizing the one they're not using so that they get frustrated and start using it. Some mice refuse and will never use one of their limbs. And I'll show you, but the control mice I'm about to show you are the ones that only use one paw. The Park mice actually ended up using both, but I think that's totally by chance. It's low numbers. But yes, there can be quite pronounced paw preferences. In fact, it's on this slide, but it's a little hidden. So here's the reaching performance for 2 Park mice and 2 control mice. And you see the success rate over time in weeks. So in week 14, left and right reaching in this Park mouse is good. And then starting at weeks 16, 17, 18, it drops. And because these are individual sessions, and it fluctuates a little bit, but they go down and down. And really, at week 24, you quite often see very, very few successful reaching movements. We did just 2 sessions with L-DOPA, and we're still sometimes it does more than other times. So this particular one L-DOPA session, the left limb was almost back to control level. So it works somewhat, and there are, generally speaking, L-DOPA treatment. They react to L-DOPA.
Yeah.
The way we injected subcutaneous may or may not always get there.
So what I remember, so this Josh Studman did this a number of years ago with just the locomotion, and there was actually sort of a long duration effect, whereas there was a clear buildup of activity over a period, I think, of about 2 weeks.
Okay.
So
by just doing it twice, we're not getting there.
Right. So actually, with I mean, it's quite striking in the figure in the paper, though it's not commented on. But there was actually a significant buildup over, I think, about 2 weeks, which is actually kind of like the pharmacology of PD.
So maybe we should do this.
Yeah,
consistently.
Yeah.
The thing is, we don't want to necessarily destroy our others. So
well, yeah, this is the trade-off.
Because
I chronic L-DOPA treatment clearly produces some kind of plastic response, at least in humans, and probably in these mice also, though no one's really studied this systematically.
And doing it at the end is meaning that we're always concerned that the mouse dies before we do anything else.
Right. Though it might prolong the mice's life, though. I mean, there's a lot of interesting things you could do with that.
Okay, so maybe we'll add a little bit more like L-DOPA.
Yeah.
So here's your question about only using one limb. So both of these control mice, one used the right limb, the other one used the left limb, perfectly over the entire period from 14 to 26, which is maybe not surprising because they're water restricted, but they never used the other limb, even though we really encouraged it in the beginning. They just didn't. And we don't know why.
When they fail, is it mostly that they just whiff, or is there a way for them to fail and like get to the water and drop it?
No, it's mostly they don't go there.
Because I think once they touch it, the water tension.
Once they get to the water,
the surface tension just holds it on the
no, it's just not. They just don't even start going there.
So the failure is more like lack of initiation in motor action, or they break contact
Right.
with the start rod.
Yes. The non-preferred side, they just don't use. It's not that it's so clumsy that it's hopeless. They just don't use it.
Are you all right, one side than the other, or is it a random?
Well, we start with random, and we built into our MATLAB algorithm. They look over the past 20 trials, and if they start using one more than the other, then we put more trials of the one they're not using in until we give up. Because we don't want the mouse. Mice can undergo a bifurcation in their behavior, where if they get too frustrated at some point, they'll cease to work forever. I don't know who's seen it in this group, but they just can completely to the level of total starvation. They will refuse to do anything any further ever if they get too frustrated at some point, or too stressed maybe. So it's a combination of stress and frustration. At some point they just give up. So we try to never have that happen, and then at some point we'll just make sure that they get enough rewards to stay motivated.
So it's like postdocs and graduate students.
Could be. And now we're getting tested on the faculty level. But that's a whole nother issue. All right. So here's a little fun side project that I just want to briefly mention. Is another honor student. She's actually doing really well. Both of them actually are going on to grad school, which is unusual because most of our students are pre-meds. So I'm very happy for our two undergrads this last year. And she did something which I thought would be interesting, is that we are looking at facial movements, which has not, as far as I know, has not been studied. But anyone working with Parkinson's patients and caregivers know that quite often hypermimia and sort of a rigid face is a problem because they can't get their emotional contacts across as well. We wanted to know whether there's any indication of mice having changed our facial movements. So we use the same exact mice in the reaching task and just point the camera at the face and then do some analysis. So here's just a video of the nose, and we use deep lab cut to track the nose, so we can see the nose tip as it moves over time. This is one of them is a left and one of them is a right movement. And you see actually deflection of the nose here. Down is left and up is right, and 0 is actually straight on. And this is a mouse that actually is a little later. I don't know why the labels disappeared on me, and it moves less. And we quantify. Oh, the labels come later for some reason. Okay. So week 17, week 26, in both cases, it's a no reach. So we compared the same limb behavior, which was a no reach trial, to the nose. And you can actually see nose movements towards the reward when there's not even a reach. You see it more consistently when there's a reach, as we'll see in a second. So this is then the maximal nose displacement to the left and to the right. So up is to the right and down is to the left. For each trial, we scored how much did they get to the left and how much did they go to the right. We had the no reach to the left and no reach to the right responses, and the reach to the left and reach to the right successful responses. And we compared, or semi-compared, how much did the nose actually orient itself towards the spout. And you see that there are actually significant differences here between controls, which is the leftmost column, and for the different movements. So if you go for no reach right, you see that there is significantly less nose movement in the Mitopark mouse for no reach right. And if you look here for the left side, we also see a significance in the reach trials. So the yellow one is the reach left, and you see a significant decrease between week 17 and greater week 21 for reach left and the maximal nose displacement to the left. So the nose movements also get hypodimensional, so it's certainly less nose movement. And I don't have enough data here about the nose movement, but you can also see that they anticipate that the nose movement actually precedes the limb movement. And we follow reaction times of the limb movement, which I also have got in slides here. And it turns out that one particular mouse was always first moving the mouse to the left. And then when the reward was on the right, it then switched to the right side with the nose, and its reaction time for right side movements was slower. So it had an internal preference to the left, but it would move to the right, but it slowed it down by first orienting to the left with the nose. So the nose readout is kind of behaviorally interesting for those who love mouse behavior in more detail. So the second thing Sammy did is she did facial motion energy, which has been done fairly extensively in the literature for some aspects of behavior. One by one. Motion energy is just, so instead of tracking individual key points and individual whiskers, you just look at pixel contrast difference from frame to frame. So if anything moves, you're going to have the same pixel hit a different part of the image next pixel. And that gives you a difference in color, in intensity from pixel to pixel. And that gives you the motion energy in these ROIs, one at the eye, upper nose, whisker pad, under eye, and one for the paw. And then you can track motion energy as a function of time in those different ROIs, and you can relate that to the behavior. And here you see actually some anticipatory movement of the actually the eye before the reaching even starts. So you see that the whole behavior is a more complex sequence of events that precede the reach in some cases. And again, this can be scored and analyzed, and you can find that the Mitopark mice here as the FME facial motion energy over time and over weeks of Mitopark development, that the amplitudes get less for both the reach and the no reach trials. So there is that reduction also in facial expressiveness with the task. So we don't have any plans right now to make the physiological study out of this in terms of recordings in the orofacial areas of cortex, but the idea would be that you might see the same things as you see in the limb area of motor cortex, and that there is changes in orofacial motor cortex just as there is in limb motor cortex. And it's a robust, I think it's a good model for that, and it might be worth pursuing at some point. All right, I want to give an introduction, and I guess I'm running a little late, so I will speed this up a little bit, but our wide field imaging. So we introduced voltage imaging for some years now through a collaboration first with Francois Saint-Pierre and Baylor at Houston, who developed the actual sensor. His whole lab is tailored to optimize proteins for specific functions. They have a platform where they can image 96 sensors in parallel in the well and look at the cell expression system on the quality of voltage sensing, or they also do ATP sensing. They do all kinds of strange sensors, and they optimize them through massive throughput and mutation and analysis. And they came out with some very good and improved voltage sensors, that transgenic voltage sensors that classically have been kind of very poor in response times and signal amplitude, but are getting now much better. And I want to encourage folks to consider those as the next big thing in neuroscience, is more voltage imaging. So here's what Yun-Miao Wang in my lab accomplished as a PhD student. So we developed a system to inject the AV vectors to get pan-cortical. So we want to do cortex-wide expression, but there's no transgenic mouse, so you have to work with AV injections. That is kind of hard to inject everywhere in the cortex. So we developed a system where we injected once in the ventricles at P0 when they're born, and the blood-brain barrier isn't formed yet. And you can get pan-cortical expression in pyramidal neurons with that. And this is just showing the extent of the expression, and is showing, actually, you see that this is a membrane-bound protein. It's also using a KV1 motif, which means it's only expressing in the perisomatic area, not in axons, because otherwise axonal label can take over. I mean, it might be good for some people, but we didn't want the remote axons to dominate our imaging signal. So it's all local cell signaling and voltage. let me not even go through the setup. We have a 2-camera system. It turns out that that all fluorescent imaging of living brain has contamination of blood flow, and you get beautiful heart. And we'll see that actually you get beautiful heart rate modulation of the imaging signal, no matter what you image with fluorescence. So we use our second camera and our second channel with what we call the red fluorescent protein, or the reference fluorescent protein, which is a red fluorescent protein. And this is not voltage dependent whatsoever. So it's just a fluorescent protein, and it's just dependent on hemodynamics. And we can then use regression to regress out the contribution of the hemodynamics on the voltage signal by taking the reference signal and use its signal to regress out that same component from the voltage signal. So until our voltage signal gets cleaned up and no longer has, for instance, these heart rate fluctuations in it. So this is just the analysis pipeline, and the paper is published. So if you are interested in this, you can certainly read this paper and see our analysis pipeline that leads to what we think is a pretty pure voltage signal, at least in most pixels, that reduces all the other artifacts. As a proof of concept in this paper, we showed that if we do a local field potential recording in a lightly anesthetized animal where you have these spikes of voltage in cortex, this is a light isoflurane anesthesia, that you get the matching spikes in the voltage imaging, but you don't get just one recording site, you get sort of pretty much a cortex-wide LFP-like recording, and you can look at correlation maps across cortex. And already you see actually that whenever you have a spike in the isoflurane animal in one part of cortex, you see a high probability of the exact opposite part of cortex that is connected to the callosum to also have a spike. So it's a lot of bilateral symmetry, and we'll see that actually during behavior, that there's a lot of bilateral symmetry, even for unilateral behaviors. So mice are very bilateral in their cortical network activation. Some of this just shows that we can see signals all the way up to electrical. We can follow the LFP signal correlations with voltage imaging up to 60 hertz about. It's significantly above chance for signals up to 60 hertz. So we can follow at least mid-level gamma oscillations with that. This is shown a whisker stimulus. So we twitch one whisker, and this is a very well-established paradigm in neuroscience for those people who study barrel cortex. And you get this contralateral barrel cortex response, and we can see a 10 millisecond response that then spreads to neighboring whiskers and to secondary whisker fields in motor cortex. And you can do this on single trial. So this is a single trial trace, and you see the individual peak of voltage in single trials. This just shows the frequency following. So we have 40 hertz or 60 hertz, either visual flicker or air puff trains. And we look at the visual cortex or the barrel cortex, and you look at just the power spectrum, so the frequency following of the local voltage signal, and you can see frequency following up to 60 hertz. Which means that at least an imposed oscillation of 60 hertz, you can actually see, and this is an average signal, but you can also see it in single traces. You see beautiful oscillations at pretty fast frequencies. This is important because you would not be able to follow any of this with calcium imaging. So that's a definite advantage, a speed advantage of voltage imaging over calcium imaging. It does have some other advantages, like it really shows us subthreshold voltage dynamics. It doesn't have to be spiking. In fact, we think that spikes are a minor contributor to this. And most of this is the subthreshold synaptic integration that we look, which the control to like cellular dynamic is an important part of what neurons do. Here is the reaching task in voltage space, and you see actually that as the mouse anticipate the reward and then reach and consume the report, that there's a very complex activation of large parts of cortex. So that by itself is kind of hard to make sense of. So we use dimensionality reduction, specifically independent component analysis. Let me not go through the details of this slide, but it's the method of independent component analysis is similar to principal component analysis, but technically a little different. So that gives us not maximal separation of correlation, but it gets us maximal independence of signals. And we can extract multiple components then out of that, measure cortical activation. And each component is identified by a unique temporal spatial dynamic. So you have this temporal dynamic defines a network where all the contributing neurons that are shown here in red, because they load with this component, are sharing that exact temporal dynamic to some degree. And when we look at the fastest response to the reaching movement, then we see this level of cortical areas involved. This is a one-sided reaching movement, but you see bilateral activation. And that is very common in what we see. If you do a left movement, the right side goes off a little more, but not that much more. So it shows that a highly bilateral structure of cortical activation reaching movements. So we also use this to, so this is a modern thing. You'll see it in almost all sort of complex behavior, mouse behavior imaging and neuropixels data. We now use neural networks that we train to see how much information we can extract about the behavior from the neural activity. And we can show that we can, with our voltage imaging, we can from 36 regions of interest, just using the trajectory of that activity in those ROIs, we can predict behavior with a high level of accuracy. We can see the stimulus response, and we can see how the stimulus response lasts beyond the stimulus until the reach. So you see how information about a stimulus is maintained and where in cortex it is maintained over time. And then you see how from the stimulus representation you build up also a prediction of the motor behavior that the animal is going to show before it's even showing it. So it's a way to extract. Unfortunately, you can't easily tell exactly how the neural network, what exactly it extracts to do that, but the information is in the activity of those pixels. We also did a frequency band specific analysis and see in which frequency bands that information is, but I don't want to go into that. It's not done in Parkinson's mice at this moment. So finally, I want to take 3 minutes to talk about Aurélie Pallas' work, which is also still in a preliminary phase, but we are using Mitopark mice, the lesions work still, and we are doing retro injections of AV label in either the spinal cord or the STN. In particular, this set of slides shows the subthalamic nucleus, and you can see that if you inject both the red reference vector and the JEDI1P voltage sensor gene in the STN, that you then get nice cortical layer 5 label, which actually spans M2, M1, and S1. So all of those areas project to the STN. Aurélie is a master in making very good glass windows that give us a clear view of cortex. The previous work was done in the Intel skull, but for the layer 5, we needed more clarity and better. And the best signal we could go, and we went for the glass windows. And this is actually not easy to do. Have a glass window over the midline is kind of hard because the midline tends to bleed a lot when you do surgeries. But Aurélie managed to do that. And then you can actually follow layer 5 activity in the Mitopark mice as they age, because we can do this for weeks on end every few days, and can follow the activity of the same neural population over weeks. And we can study what changes in their activation. She very carefully aligned the different mice to the same Allen atlas to get the ROIs that match the S1, M1, and M2. And then she looked at the trajectories of activity. Now, this is maybe mostly a negative result, but it does show you the extent of activation of STN projecting neurons in the cortex prior to reaching movement, reaching from S1 to M2. So it tells us a fair amount of what the STN receives in terms of information, and that's also interesting and relevant to Parkinson's disease. But it turns out that the progression in these mice led to very little change in the intensity of the signal. So that seems to mean that STN projecting neurons are not in any major way impacted in their function. We're doing more mice. If anything, the one mouse that did show changes is an increase in activation, which is the opposite of what happens with just a window going bad or the dye getting bleached. So it seems a real physiological result, but it's actually an increase. So if you look at the light green, that's 23 weeks, and the black is 15 weeks. If anything, there is an increased signaling in SDN projection with progressive metaphor, which could be a compensatory change of some sort, but it's not the one that is sort of healing over and not signaling anymore. So that might be the outcome of this study. At the end of the day, that leads me to my conclusions, which is that mitral mice are a robust model of progressive Parkinsonian motor deficits. Later stages are characterized by a lack of movement, and I didn't emphasize that, but if you looked at the reaching movement statistics, they have very few reaches at the end, and most of that they just don't move anymore. And it kind of looks like they want to, but that's hard to quantify. Step cycles slow down by increased stance duration. Swing phase is largely maintained, and they just have many fewer bouts of moving. Again, initiation seems to be a big issue. We think that there is some reduction in EMG burst amplitudes and delayed timing is what I want to say here. But we need more mice. Hypomimia, meaning that there's indication that the facial movements are just as reduced as the limb movement, and that could be interesting in the future. Voltage imaging shows that STN projecting neurons are first active with relation to reaching movements in the widespread areas of cortex, but not really showing a change in getting smaller over time. And reach activity is generally bilateral organized, and again, for the SDN projecting, we don't see a deficit with progressive metaphor. That's it.
Okay,
I want to acknowledge all the folks in the lab that put in the hard work.
thank you. That was fun. So any questions or anybody on this? Anything in the chat? So, okay. So Peter, you're looking at other, but you said you injected not just the STN, you injected the spinal cord.
Yeah.
You're looking at pyramidal tract neurons also. You haven't.
Correct. There's only one mouse each at the time.
Right.
We are actually also, we've done more injections. Well, not we hourly have done more injections. We're collaborating with Yoland, actually, who's done more in the primate. And there's a grad student in Yoland's lab to just looking at the collateralization of these neurons in the other brain areas. So there'll be a little paper about that.
Yeah.
To compare primates and mice in terms of collaterals of corticospinal neurons.
Right. What about IT versus PT neurons?
Well, those are all ET neurons.
Yeah, these are all PT neurons.
So that we don't know anything about IT neurons. So that would be a separate.
That's a separate project.
That would be a new project. There are ways to label them genetically and otherwise. It's just a question of how many people can pay from the real brand.
Right.
It's not so many.
Yeah.
Curious about the EMG data. Do you think of that as fewer motor units being activated or weaker activation in the same motor pool?
Would we know? Well, the variability and the broadening seems to be something that would be indicating weaker and more variable activation. And because we're thinking that the electrodes stay put in the same place, we're more or less, I mean, we can't say we're recording from the exact same neurons over time, but my feeling it's that the same units are getting activated less. It may also mean that some drop out and are no longer activated much. So, but it's not. I wouldn't say that it's the cessation of the ability to. I don't think the lack of initiation isn't just that they can't get the EMG active enough. I don't think you'd see when it's kind of hard to say because when they're not moving, how do you align to a step cycle that isn't happening? But I think that there is not the lack of initiation isn't just a lack of motor neuron drive. I think there's a whole state dependent, just not moving state that is independent of this change of EMG.
Yeah, I'm just trying to conceptualize that they're changing the excitability, motor neurons, or if the inputs are getting weaker, or fewer inputs are being activated through the motor neurons themselves. Yeah, at what stage, I guess, you're trying
Yeah,
to.
well,
conceptualize
the-
the feedback of problem.
Well, we want to record from those neurons in the cortex, and,
Sure.
and we can at least let you know what the correlation between cortical firing and that is. And there are steps in between. So like you said, it could be that something happens at the level of spinal cord. There's dopamine input to the spinal cord,
Right, but
So-
mainly from the hypothalamus.
right? But this mutant will hit
It
those.
will hit those, yeah.
So it's not. It certainly could
Well,
be
we-
something. One should be aware of that as,
Yeah.
as a
So
possibility.
what typically happens in PD, I mean, this was described decades ago with simple movements. There's typically the size of the agonist burst is smaller. So this could be somewhat.
Right. But then even in patients, you probably it's hard to say exactly what happens at the level of motor neuron
Oh,
output.
absolutely. I'm sure somebody's, I mean, there's some smart motor physiologists who probably looked at that, but I can't. I will say though that, initiation of movement is a deficit. That's a prominent PD feature. And, I would say quite reliable.
Yeah, so it might be the best model of initiation.
Yeah.
Unfortunately, for doing imaging studies, when there is no initiation,
Initiation,
we
right.
have nothing to see. So
Yeah.
it doesn't work in that way.
All right, well, thanks very much. That was fun.
Yes.